My background for this is actually, I'm almost not a biologist, but came into marine microbiology about 30 years ago. And I've been trying to, so then I was there when the viral story started in microbiology, about 25 years ago. And or the part of this that I'm talking about here is how to incorporate the viruses into our understanding of how the surface ecosystem of the ocean works. And as I said, there is about 25 years story, because when I started, the textbook said that viruses cannot be important in this environment because there are too few hosts there. And people did not realize that the number of hosts had increased by about 3 orders of magnitude about 10 years before this, when the fluorescence microscope came into use. And since in these 25 years, there has been a huge improvement in our observational and experimental work in this field. I'm not going to review that. Jed Furman gives the next talk, and I think he will give you a lot more on the historical details of this part. This is where I have been working, where we try to understand the role of the viruses in this ecosystem. And to sum up the conclusion, I think the state is not particularly impressive, because you can ask very simple questions about this ecosystem, like what controls the viral abundance in this system, or how is the diversity of the host linked to the viral activity. And I would claim that there is no clear answer to these simple questions at the moment, still after 25 years of work. And to explain this, I have to make it very simple, go through the very simple idea of top-down control in an ecological system. And this is then an example with a predator and a prey. So if you have the prokaryote community and you have protozoan predators that are non-selective, if this system is in steady state, there has to be enough prokaryotes for the protozoan predators to grow as fast as they die. That's the simple argument. That's the steady state argument. And this comes out, if you do the mathematics and look at the parameters of this, you actually come out at around 10 to the 6 per milliliter. So this seems to fit the observation, and also the observation that this 10 to the 6 per milliliter is relatively constant. This would fit the idea that there is a top-down control. So whether you change the resources, the predators then respond very quickly and keep the population at the same size. Exactly the same arguments you can use for viruses. Just that then the size of the prey group will be much smaller because they have a much higher, the absorption constant is much higher than the clearance rate for the predator. And an important conclusion of this simple argument is that the size of the prey community, if there's a pure top-down control, is independent of the competitive abilities of the prey. So how many you have of an organism does not really depend upon its competitive ability. It depends on the top-down control. And the importance of this, you can see if you take this relatively recent discovery of the SAR11 viruses. The SAR11 is probably the most abundant organisms, at least organism in the ocean. And it was argued that this was probably because it had high defensive abilities. No, it turned out that it actually had a lot of viruses. And then the argument was that then it's so abundant because it has high competitive abilities. But then we get into a problem. There is sort of something not quite consistent here. how can successful competition give high abundance if there are a lot of viruses, so we assume it to be top-down controlled. There is something not really consistent here. So the models we have used before, we have termed them killing the winner, is simply a construction where we combine different top-down controls. So this is the predator control, and then if you add viral control of this, you can have host groups within the prokaryote community, each host group controlled top-down by a virus. So then you get a simple theory for diversity. It's simply how many small boxes can you fit within the large box. So it's the ratio between how small populations the virus control gives to the size of the control given by the protozoans. Very simple. And it actually also suggests the mechanisms for the control of the abundance of viruses. Because in this model, what viruses do is to compensate for the differences in growth rate of the host groups. So if all host groups grow at the same rate, there will be no viruses in this model. If there's a large difference in growth rates, you get a lot of viruses. So the viruses, so you get kind of hen and egg system where viruses keep up the diversity within the community, but it is the diversity within this group that determines the amount of viruses. Now you can add a trade-off in the host between their competitive and defensive abilities. Then you get another interesting aspect here. You get the competitive hosts can grow rapidly because they have a low cost of resistance, but they become rare because of the strong top-down control. While the defense strategists become abundant but grow slowly. So you get an opposite distribution of the host abundance and the virus abundance. The abundant viruses correspond to the few but rapidly growing hosts. So this was sort of the state of the art. And but with the SAR11 story, you can see you get a problem. If you put the SAR11 as a competition strategist, you get a problem of the abundance of the SAR11. If you call it a defense strategist, you get a problem because it seems to have a lot of viruses. So the model doesn't really seem to be able to reproduce the observations. And as a general thing, even after 15 years after publication of this model, there is not really any hard evidence either supporting or refuting it. I remember Jed Furman was a reviewer when we published the original paper, and he said he was moderately enthusiastic and said that I'm sure the world is more complex than this, which of course is true. And here's my suggestion for at least one of the problems with this, looking at the world in this way. It may be these host groups. Because if you immediately assume that the host groups correspond to species, then you say that the viruses are species-specific. Now, if you do the other extreme and say that the viruses are strain-specific rather than species-specific, you get something interesting here. You get a three-layer model. You still have the community size controlled by the protozoan predators. Then you have the species, and then you have the strain size controlled by the viruses. So you have predator control on community size, and you have virus control on strain size, but you have lost the control on the size of the species, or the explicit control. And that's the level where most of the data exist, and where people try to compare the models to the observations. So you can do this, what seems like a trivial statement here is that no, the individual per species in such a model is the individual per strain summed over all the established strains. That sounds like an interesting self-evident statement, but actually the consequences are quite large. So if we start by doing slightly more than this one virus, one host model, and look at one species, and then try to do this chemostat thing of infecting one host in the chemostat. So if we start here, this is the traditional chemostat, where your resources from the reservoir goes into the host population. So here the resources sits in the parent strain. Now you infect by a virus, you get something like this, with a low population and free resources in your culture. So you move the resources back to the free nutrients. Then this creates a niche for a mutation in the host, because there are free resources in it. So what you would expect is this thing, which is actually what many of the experiments show, that you get a system dominated by a resistant host. And the resources now has moved back, but not into the susceptible host, but into the resistant one. So the free resources, in a sense, moves like this. Which means actually that theoretically there is a kind of mature state of these arms races, where all the free resources are gone. They now sit in susceptible hosts. So of course there will be evolution from this stage also, but there are no free resources anymore. So at least I would suggest that you would expect a much more rapid evolution until you reach this mature state. And as a kind of free bonus from this exercise, you can solve what in this field is called Weinbauer's paradox, because Marcus Weinbauer wondered why is there such a difference between natural system, which looks more or less like this, where you have most of the hosts are susceptible to viruses. And if you try to do this in a laboratory, and you immediately get a resistant host. In this model, this is just the early and the late stage of the arms race. So what happens in this chemostat example? In the initial case, with one strain, the original strain has to grow equal to the dilution rate. Then if you get a virus, you get a new, when you get into the situation with a new strain and the first one controlled by viruses, the one which is resistant is the one that has to grow with the dilution rate. The susceptible one has lost both to dilution and to viruses, and therefore has to grow faster. So you move, as you get more and more strains here, there is a curve for each strain, you get a higher and higher concentration. This is theory. You get higher and higher concentration in your chemostats, more and more strains, and the the first parent strain grows faster and faster. So actually, now if you put two species in your chemostat, you immediately see all these strains can establish. Now you can establish strains from different species in the same chemostat, as long as they can grow faster than the dilution rate at the concentration established in the chemostat with all the viruses there. So you can get more and more species in your chemostat. And as a hypothetical example here, you can get a situation like this with all these different species and all these families of strains below here not shown in this figure. So you can get a very complex, in theory, a very complex steady state in your chemostat. Many species, many strains. And how many strains you establish from each species is determined by its competitive abilities. This figure determines how many species you get there and how many strains. But it does not determine how many individuals you get of each strain. That is determined by the defensive abilities of each strain. So now you suddenly have a system where your abundance depends both on your competitive abilities and your defensive abilities. If you look at one species with this model, and these are the different strains, the more they defend themselves against viruses, the higher the cost of resistance, so the slower they grow. And with this model, because there is a trade-off, you get more and more individuals for each strain. So each species is actually dominated by the slow-growing strains here, in terms of numbers. And you have a few fast-growing competition strains subject to viral lysis. So many strains require high competitive abilities. Many individuals per strain requires high defensive abilities. And high abundance of a species then requires both. So now you have a theory that where abundance depends both on your competitive and your defensive abilities. So this is then just a model community where the parameters, the defensive and competitive parameters of each species is drawn randomly. Then you can get things like this. And interestingly, you get this thing, here's the growth rate, but the community is dominated by these slow-growing strains. Which actually is interesting in marine microbiology, because you have all these, when you observe the system, you have a few active strains and a lot of low active strains. So there is a long debate in marine microbiology about dormancy in this system. So this actually suggests a reinterpretation of dormancy, where the organisms are dormant, not where they are starving, not because there is no nutrients there, but perhaps because they have closed the gates to keep the enemy out. Little like an old medieval town, siege town. And to become abundant in this system, there has to be a low trade-off. This is just taking these randomly generated species and looking at the trade-off. And you can see how this model then gives, there's a logarithmic scale here. So there are a few species dominating, and it's because it's those with a low trade-off between competition and defense. So becoming successful in such a system is not a question of being defensive or competitive. It's finding a mechanism that has a low trade-off. This last one was just to show that you can actually combine this with a more ecosystem model, where you can link the position of this line in your graph to the position of this line in the graph to the abundance of the predators in the system. So you actually can get a system combining the diversity with the ecosystem structure. And I think the conclusion is that to build a realistic model, one needs the seeding community here. And the intriguing aspect of generating a seeding community is not to experimentally determine that all properties of all possible organisms in the ocean. It's more to understand the trade-offs. So why should extreme minimalism in SAR 11 be so successful? Is there wartime production of viruses? US Liberty ships or German Tiger tanks? Are there RISC or CISC processors? So, thank you for your attention.